Hello everyone, welcome back to Channel for Doctors. Here today we are going to continue a section of the microbiology. Today we are going to study about catalyst positive organisms, those organisms which uh, inherit the enzyme catalase. Catalase is an important enzyme for degradation of hydrogen peroxide into the water and oxygen bubbles. So those organisms which carry this enzyme actually are prevented from the injurious effects of the hydrogen peroxide before it can be converted into the myeloperoxidase with the help of the enzyme NADPH oxidase. So what happens whenever there is an infection with a bacteria, the phagocytic cells will come and kill it. So the phagocytic cells contain H2O2, which is hydrogen peroxide. This H2O2 is converted into the myeloperoxidase with the help of enzyme NADPH oxidase. NADPH. So, NADPH oxidase is an enzyme which converts hydrogen peroxide into the MPO, and this MPO in turn is toxic to the bacterial life cycle. So those people who have deficiency of NADPH oxidase, they are more susceptible to the bacterial bacteria which produce catalase, because catalase in turn hydrolyzes H2O2 into the water and into the oxygen. So it cannot be converted to MPO, and bacteria will in turn survive longer than the normal duration and will cause chronic granulomatous infections. So this disease causing organisms basically have the characteristic feature of producing recurrent infections with certain catalyst-positive organisms. Examples including the nocardia, the Staphylococcus, Rickettsia, Candida, Listeria, E. coli, Burkholderia, Pseudomonas, Aspergillus, H. pylori, and Bordetella pertussis. So these are all the catalyst-positive organisms, and these organisms produce the recurrent infections in those people which lack NADPH oxidase. And then we have some special pigment producing bacteria such as the Actinomyces israeli, which produce the yellow sulfur granules that are composed of the filaments of bacteria, and that's how you recognize them on their blood cultures. Then we have the Staph aureus that produces the golden yellow pigment. Pseudomonas aeruginosa produces the blue-green pigment, and then we have the Rickettsia, which produces the red pigment. Then we have in vivo biofilm producing bacteria, which includes the Staph epidermidis that causes the catheter and prosthetic device infections. Then we have the pathogen streptococci, which includes the strep mutans and strep sanguinis that produce the dental plaques and infective endocarditis. The Pseudomonas aeruginosa cause a wide variety of infections, but the most common being the respiratory tree colonization and cystic fibrosis and ventilator-associated pneumonia. It can also cause contact lens associated keratitis in those people which wear contact lenses for a significantly longer period of time. Then we have non-typeable and encapsulated H influenza that causes the otitis media. So, and then we have some spore forming bacteria. These spores are basically the protective mechanism of bacteria when they want to survive or when they survive the harsh environment, or they want to resist degradation. So some gram-positive bacteria can form spores when nutrients are limited. The spores lack the metabolic activity and they are highly resistant to heat and chemicals. The core of these spores contains dipicolinic acid that is responsible for heat resistance. Must be some foods and instruments must be autoclaved to kill these spores, as is done in the surgical equipment by steaming at 121 degrees centigrade for at least 15 minutes. The hydrogen peroxide and iodine-based agents are also sporicidal. The examples of spore-forming bacteria includes Bacillus anthrax, Bacillus cereus, which causes the food poisoning. Then we have Clostridium species such as Clostridium botulism, Clostridium difficile, Clostridium perfringens, and Clostridium tetani. And the disease and pathogenesis caused by these organisms will be studied in the subsequent sections. And then we have some important bacterial virulence factors such as protein A that binds Fc region of the IgG and prevents opsonization and phagocytosis. Protein A is an important pathogenic and virulence factor of the staph aureus, so A for protein A and A for aureus. Then we have IgA protease, that is an enzyme that cleaves the IgA, as the name suggests, allowing bacteria to adhere to and colonize the mucous membranes. It is secreted by three organisms: Strep, pneumo, H. influenza, and Neisseria. Then we have the M protein that helps prevent phagocytosis also. It is expressed by group A Streptococcus. Sequence homology with human tropomyosin and myosin, which is also called molecular mimicry, can possibly cause the autoimmune response as an acute rheumatic fever. So what happens in acute rheumatic fever is that the antibodies that are produced against the M protein of the bacteria will also attack the M protein that is present on our myocardium, which will result in the autoimmune acute rheumatic fever. So about acute rheumatic fever, the pathogenesis and disease progression, we have studied the cardiology videos. In this session, we conclude the bacterial pathogens factors and the catalyst positive organisms, the spore forming organisms, and in vivo biofilm formation producing organisms. So this was all about today's video. If you have any question, you can ask in the comment section. Thank you so much.